Good morning, everyone. My name is Louis Cobble. I'm one of 6 apiary inspectors in North Carolina. It's September 14th, 2023, and with me today is Dr. Frank Rinkevich, research entomologist at USD ARS in Baton Rouge. Frank, how are you?
I'm doing very well, doing very well. Just got back from Apimondia in Santiago, Chile, and it was a very interesting meeting because we got to see what beekeepers and bee scientists from all over the world are doing. And there's going to be a lot of things happening differently in the beekeeping world because with the arrival of Varroa in Australia, that's really going to play a big factor in how bees are shipped around the world because Australia was the last Varroa-free bastion of beekeeping in the world. And now that they got it, I think there's going to be significant changes coming out of that. One of the things that I was obviously interested in was Varroa mites, and people are noticing resistance. So we found out that yes, especially to Amitraz, it's in Spain, it's in France, it's in Argentina, it's in many countries. So it's interesting to hear a lot of feedback from after my talk from people saying, yeah, I noticed Amitraz doesn't work like it used to. So this is something I think that is a global issue as well. Found out that there are different ways that they can become resistant. So this was very interesting. Talked to a lot of people from Spain, Joel Gonzalez Cabrera and Sarah Marino Marti and Sarah Hernandez Gonzalez also was really interested to see what they've been doing in Spain. So this is something that really invigorated me to do some really new research. Saw a lot of new products out there, most of which are not registered in the United States that contain Amitraz. Saw that Amiflex was out. Hopefully get my hands on some of that now, just to do some testing to see what's going on with that. And I just think that there's some changes in the marketplace for Amitraz, but I think that there's also being a shift towards, by beekeepers especially, to use things besides Amitraz because they're recognizing it's not working like it used to. So I think we are seeing that behavioral change, especially here in the States.
Yeah, that's good.
Summer.
Yep, I think that's very good. I think we have been a little too reliant on that one product. That I guess it's, as you mentioned in your paper that we'll talk about here in a little bit, it's easy and it's been effective, and so that can lull us into bad stewardship practices. And I think it may have caught up with this. But so you didn't just attend, you spoke at Apimondia.
Yeah, I presented. I only had 15 minutes to talk about 5 years of research, but I did squeeze a lot into my talk, and I really focused in on that we do have resistance here in the United States, but there's also a lot of populations that are still susceptible to Varroa. I thought that was kind of it's really important to tell about the whole picture because I think people think that it's everywhere, and not all mites are resistant. And I think,
right,
and I really need to put some data to this, it seems like overuse and over-reliance on Amitraz is the only thing you do to control Varroa. Seems to be what's really driving this whole system. When we talk to beekeepers who use other products in addition to Amitraz, they don't seem to have the problem. When they follow the labeled instructions, it seems to, Amitraz seems to, resistance tends not to develop in their apiary. So what the really cool thing about this is that we know how resistance develops from all these other great examples in other agricultural systems. Looking at corn rootworm, for example, the anything that parasitizes, like we have so many great examples of this. So it's surprising that it's not more widespread with Varroa. So this is actually kind of so we know that there's other examples of resistance, but there's also examples of how we can overcome that resistance from those other systems. So hopefully we could borrow some of those ideas and apply it. And one of the things that's pretty simple that we can do today is rotating miticides.
Yeah. So I have some questions about all of that as we talk about your paper that was published in March. You're looking at, I guess, the genetic differences between susceptible mites and resistant mites. So Tell us a little bit about that. So just kind of an overview of the paper.
Yeah, so resistance is the genetic ability of a population to withstand a concentration of an insecticide that would cause mortality in a significant proportion of the population of an otherwise susceptible group. So with that in mind, the key point of that is genetic. So there has to be an ability to be inherited. Because we could see treatment failures due to misapplications, underdosing. It's 2 strips of Apivar per chamber instead of 1, and you have to leave it in for 42 days, just like the label says.
Or if it's not placed in the right place in the colony, if you put 2 strips in the honey super, all bets are off, right? So there's other issues with failures.
Certainly. Yes, exactly. And that's why it's so very important to read the label. Label is not just made up, it's there for a reason.
Yes.
What we're finding out, though, is that with those sort of treatment failures because of misuse are very different than correct treatment, but the product failures. And that's the basis for resistance. And so there has to be a genetic component to this. And so we know how amitraz works. It works by binding to this receptor called the octopamine receptor. It looks like octopamine, and when it binds to that receptor, it causes paralysis and ultimately death of the Varroa. But what happens is with Varroa that have the resistance mutation, the receptor changes so that amitraz doesn't fit into
Doesn't
it.
fit.
And that's what we've seen with many other, this is called target site insensitivity. This is common throughout every other sort of insecticide insect system, well, arthropod in the broad sense. But not too much work has been done on octopamine receptors because amitraz is kind of a very fringe molecule. It's the only representative that of the formamidine class that's actually used. Most of them aren't because of other issues with these products. But with amitraz and octopamine receptors, there's not much known. So when Joel Gonzalez Cabrera from Spain published a paper where they looked at specific mutations in those octopamine receptors in mites from France, they found that there was this one that was always associated with resistance. And they knew that it was associated with resistance because this was after mites that survived a treatment of amitraz. So they knew that they would be resistant. When they looked at it for the United States, they found a different mutation, but I wasn't too convinced that it was, I believed it was. It was very likely that it was associated with resistance, but it was based on beekeepers' observations as opposed to a quantitative evaluation. And so we wanted to make sure that the numbers were accurate. So what we did is I went back to my freezer and found mites that were quantitatively phenotyped for amitraz resistance. So we know that, yes, these survived the cup treatment. They survived the cup, right? And so I like it. So what we know that they were resistant. And so when we redid that analysis, we confirmed that, yes, this is definitely associated with resistance. The odds of it being associated with resistance are pretty high. And so I'm very convinced that we have this, it's called the Y215H mutation. All that really means it's shorthand for saying that there's a mutation in the specific part of that receptor that is associated with resistance. Interesting that we found at Apimondia. Actually, I'm not going to talk about it because it's not published yet, but
Okay,
there's some other
so
cool stuff
I
going
guess
on.
the part of this that I think is pretty interesting is that there's the mutation for resistance in the United States is different than the mutation for resistance in France. And is there any indication that there might be other mutations in other geographic locations, or we think this is the extent of it, or we don't know?
I would be very surprised if this was the only mutation, because arthropods in general, when they evolve resistance, they kind of do it in many different ways. So this mutation that we have here in the United States called Y215H, we see it all across the country, right? But what's really interesting is that we don't know if it happened in one spot and then kind of spread out from that one spot, or did this population evolve it independent of this one, independent of this one? And based on my previous experiences with insecticide resistance in houseflies, that is the case. There are these independent selections of resistance. So they're all selecting that same mutation independent of ancestry. So that's what's really interesting, is that I think these mites can evolve resistance quite rapidly. So I think what I want to know is, can we prove that with DNA data? And yes, that's what we're working on. Actually, Arianopoulos in the office next to me, he was running this huge genomic analysis that we just got back from 500 individual genomes from Varroa, and we're comparing resistance susceptible. So we're trying to find these molecular markers for resistance. And so if that's the case, we could find out, okay, is this arose once and spread, or is it independent evolution? I would bet it's independent evolution, because one, we see that in other insects, but also in arthropods, let me say that. But also two is we see that resistance tends to be restricted within operations. So beekeeper A could have resistance with Varroa, but beekeeper B, 3 miles down the road, has completely susceptible. So I don't think that I think Varroa populations are kind of isolated based on beekeeper. within beekeeping operation, because they move things around and they share equipment between yards, within the operation, I think we see similar genetic Varroa populations, but in different beekeeping operations, we don't tend to see that unless they're sharing equipment.
Right.
So
I guess what is I think this was kind of before my time, but when we observed resistance to coumaphos or thofluvalinate, it was kind of widespread, and it was just, okay, that's all she wrote on that. But the trend seems to be different with amitraz. Like it's in pockets, and also can we regain susceptibility in populations of mites if we are better stewards?
yeah, that's actually something that is interesting. So for fluvalinate resistance, that's been pretty well documented in Italy. When they found resistant Varroa, they didn't treat for 2 years, and susceptibility was restored. In fact, Sarah Moreno Marti, who from Spain, but works with Joe Gonzalez Cabrera, she actually did that study. They started off with these populations of either they were resistant to fluvalinate, and they split them into 2 groups. One they treated with fluvalinate, and the other they didn't. And with over the course of 2 years, ones that were not treated, susceptibility came back.
Oh, okay.
So it revert. So we can restore susceptibility. However, we don't know if that's just a population displacement or is it a genetic reversion. It's a little tricky to know exactly what's happening there because genetic reversions are difficult to detect. They tend to just be population displacements. The only way that we could check that for real is to sample those genetics and see if, in the broad sense, does the whole genome evolve, or is it just are we getting immigration of other mites that kind of dilutes out those resistant ones?
Yeah, I guess
so. That's kind of, you know.
I guess I'm not a very smart fellow, and certainly not with genetics. But
oh, don't sell yourself.
It seems. I was just thinking about the genetic diversity in Varroa mites, and I'm looking at their family tree, and it's more of a stick. Like, so everyone's the father is also the uncle, right? So how does that play into all this?
Yeah, so actually the Varroa reproductive system actually is a great way to move these for resistance to evolve and spread very rapidly.
Yes.
Because if you have, like I said, that line, mother, brother, sister, and so on, like I said, that vertical family tree, it's almost like it's semi-clonal, it's full sibling mating. It's not clonal reproduction like you get with an aphid. But what happens is once that mutation springs up, all of her descendants will have that mutation. Not half, like if you had like outcrossing between other individuals, but because of that, somewhat linear vertical mating pattern, that whole lineage from that point has that mutation. So that's where it gets really, really difficult. Once it appears, it tends to spread very rapidly. And that's what we've been seeing across the board. I mean, a few years ago, when we first started hearing about resistance, now we saw it evolve very rapidly. And then also too, we've seen it happen in many other places where nothing, and then all of a sudden it takes off. So I think it's that strict reproduction that doesn't, I think what happens, and I don't know this yet, but I think Varroa are very good at producing genetic mutations from generation to generation, because with that vertical reproductive, that very vertical family tree, in order to have genetic variation, you have to have high mutation in that case, because you're not having importation of other material. Unless so heterozygosity is very low in Varroa populations. So yeah, that is definitely a big thing with it. That's what makes them probably more likely to evolve resistance. And there's a whole list of these biological characteristics that are ideal for originating and spreading resistance, and Varroa kind of fits almost every single one of them.
Cool. I was thinking about I was reading the paper, thinking about, so you collected what we thought were resistant mites and what were susceptible mites, and you started doing this testing, and you're finding susceptible mites in the what you thought would be the resistant group. But it wasn't.
No, the opposite. We found genetically resistant mites that looked
Yeah,
susceptible.
right. In the susceptible group. Yeah.
Yeah,
So
yeah.
so did you know what was going on when you first saw that? Or did you were like, oh, maybe this test isn't what we thought it was, or how did that go?
Yeah, so you always question how you might be wrong. And so what it comes down to a bunch of things is that if you have a genetically resistant mite appear susceptible, there has to be something with the test. And what we found out was that, okay, before I get down that rabbit hole, I just want to flip it the other way, is that if we have a mite that has a susceptible genotype, so if it looks susceptible with the DNA, there's a very strong probability it's going to be susceptible in the test. If it has a susceptible phenotype, meaning it falls on that tray, there's not too high reliability that it is actually genetically susceptible. So we could get genetically resistant mites appearing susceptible in the test. If they look resistant in the test, then they have a resistant genotype. So that's where we're at with this. So how do we get a genetically resistant mite appear susceptible in the test? It's temperature, because at higher temperatures, the mites just naturally fall off. And in fact, when I went back and reanalyzed the data sets from that paper, so whenever we do these tests, I always record the temperatures. And we found out that there was an association of mismatch between genotype and phenotype when the test was conducted at a higher temperature. So typically above that 30, close to that 30-degree point. So whenever we're running this test, we have to make sure that we're recording the temperature, because that way we have to correct for natural mite fall that occurs at those temperatures. So that's where we're at. So with that being said, is that if we have these genetically resistant mites appearing susceptible in the test, and we're basing our assessment of resistance on how many mites are on the tray and how many are on the bees, I think we've been actually underestimating resistance if we get these resistant Varroa to fall due to temperature. So that really caused me to go back and have to reevaluate some of my old data, because I collected all these, every time we run this test, if we had Varroa, I usually collected them. So I have them all in my freezer. So if we go back and genotype them, we could get a better assessment of that looking backwards.
Cool. And there's a few that go the other way, right? So they just mechanically cling to or they're kind of hidden on the bee and are not exposed enough to fall. So it's not perfect, but it's pretty good.
Or they get stuck in the screen, or sometimes, I don't know why, but some of the containers, they might stick to the side. So whenever you're going to go wash them, make sure. And so, yeah, it does happen. It's not 100%. I will admit that the test is not 100% accurate in discriminating resistant susceptible. But if you look at the totality of the data, because we did 125 of these samples, and when we run the statistics on it, the probability of those things happening are extremely highly statistically associated.
Yeah, I think it's such a nice, easy, informative test. It really helped me when I was doing it in my area for folks who said, hey, I just finished treating with Apivar and I didn't get good results. And we could do this, just really just a 3-hour test and see, get a good idea of what's going on. So I think it's I appreciate this very easy to do and very informative method. So thank you.
I will.
Like
I admit the test isn't perfect, but it's very, very good.
It is very good. Yeah.
But what we've been finding out though is whenever we find high Varroa populations, they tend to be resistant. And this is mostly when we're looking at, most of all the 1,500 samples I've collected over the years, most of them have been from commercial beekeeping operations. And when we do that, we tend to see, if we find lots of mites, they tend to be resistant. And that makes perfect sense to me, because what that tells me is that those mites are escaping control from amitraz treatments. So that kind of makes perfect sense. So if you're using amitraz and you still see high mites, then yeah, they probably are resistant.
Yeah, well, I think that's go ahead.
And in fact, that's kind of what how we got put together on this, because that was an observation that we discussed very early on in this project.
Yeah. So I saw your interview with Umberto Boncristiani about amitraz resistance, and that's the first time I had seen you, and I was like, he's describing exactly what I'm seeing, and I reached out to you from there, and
that's what's interesting, is that so many times when the beekeeper says, hey, the treatment didn't work, and I go out and run the test, it tends to be correct. So
Yeah.
those observations by beekeepers are usually supported by the data.
Yeah. So I guess when I was reading your paper yesterday, I was also thinking about how useful this genetic test could be. People are asking me, what might be my best treatment? It's like, well, they're all pretty variable. I like Apivar if your mites are susceptible. And I don't know if your mites are susceptible or not. And so I think this could be a pretty good tool for apiary inspectors that could sample packages in the spring, packages from this distributor or that distributor, and try to characterize those mites to be able to make more solid recommendations on treatments. So I think this has a lot of value.
And so that's actually one of the other really benefits of doing this test is not only do you get your resistance status, but you get your mite infestation levels as well. But then based on that resistance test, it tells you what treatments might be efficacious on those Varroa. So we have lots of data showing that when they look susceptible in the test, you could use Amitraz or Apivar with high degree of confidence, knowing that it's going to knock those mite levels down pretty quickly. When you use it according to label, it'll get those mite numbers down at the end of the treatment. And we have a lot of data that support that. We don't have a lot of data, though, to support what happens when you have Amitraz-resistant Varroa and you use Amitraz, because two things happen. Beekeepers say, well, I'm not going to use Amitraz because if you're telling me it's not going to work, why would I do that? So that's case number one. Case number two, if I try to convince them to use it, they'll say, hey, are you going to pay for my colonies when they die because I use a product that doesn't work? So I'm not going to tell a beekeeper.
Right.
I would love to have that data, but also too, I need to pay them for their colonies that potentially could be lost because of this. So I don't have any data. I don't have a significant amount of data in that direction just because of the potential colony losses that can occur.
Yeah. All right. Anything else? Let's see. I think that's the stuff that I wanted to cover on that. But anyway, yeah, thank you for all your good work on this. I think super important topic. I think it's the most important work that's happening at the moment. So thank you for leading that charge. So,
well, of course, I couldn't do this without you, Lewis, because you've been my partner in crime and all this kind of thing. So it's been really great. And so just getting the word out there, because the more that we get the word out, I think that, like I said, this summer I started seeing people using things besides Amitraz. That made me pretty happy, and it made me happy in the fact that the word's getting out and people are realizing it. But it was tough to find any Varroa this summer because people are doing better treatments and better managing their Varroa. So I think that this is something that we have to keep hammering the message home and keep making sure that people are rotating the miticides, using Varroa-resistant stocks of bees, and using other IPM measures to minimize Varroa infestation. Because to me, Varroa is the number one problem. All the other things are important for sure, don't get me wrong, but I think that Varroa is such the most common factor across all beekeeping operations that this is something that is applicable to almost every beekeeper.
yeah, I think all this underlines the importance of staying on label, like using registered products according to the label and being good stewards of the product. This is an important product to the industry, and it's important that we not squander it.
Right, exactly. And that's the tricky thing with miticide development is that there's no miticide in shining armor coming to save us.
Right.
So we have to, because most miticides are just old materials that have been sitting on the shelves at other companies that they just kind of develop. None of these products are built from the ground up to control Varroa. So there's nothing really coming along the head. So here in the United States, that means we have Amitraz, oxalic acid, formic acid, thymol, beta acids, and a few other things. So we have to make sure that whatever formulation we're using, we can maintain those, because there's not anything big, unusual, different, cool that's going to be highly effective that's going to be here tomorrow. Registration takes a long time. Validation takes a long time. Finding out what conditions of these new materials work in is very difficult. So we have to make sure what we have, we can use what we have for as long as possible, for sure.
Yeah. So as someone who studies resistance, should I worry about just only using apibioxyl?
So yeah, so resistance can evolve to any of these products, but the most important thing is that the duration and intensity of selection. And I think that's what happened with fluvalnate and Coumaphos, is that once those materials were put into the colony, they go in the wax and stay there forever. I mean, I could still find Kumafos
Yeah.
in comb that has been applied in 20 years. So that's kind of the issue. So there was that constant, intense selection. That's why I think that those compounds evolve resistance so rapidly. With amitraz, it gets in there and it does stay around, but not to the intensity and duration that fluvalent Kumafos. So it's kind of a little bit less. When we're talking about the organic acids, especially like oxalic, it really is only in the colony while that strip or the vapor or the dribble is being applied. So you get this intense spike of selectivity, but it's very short. So because of that, there isn't a high pressure to evolve resistance. But I'll tell you what, you go out there and you apply oxalic acid every day, you keep coming back and hammering them and doing something like that, it can select for resistance. So I've been hearing people say, oh, resistance to oxalic acid will never evolve. I'll do it just to show that, yes, it can happen,
Right.
because whether it's oxalic acid, amitraz, or fluvalent, anything with enough intensity and selective reproduction, what's it called, with a difference in reproductive outcomes based on that genotype, it will lead to resistance. So whether product we're talking about, just being rotating and being judicious and following the label is very important.
Yep, with all the products. Yes, thank you. So I guess you have some other follow-up studies, like looking at temperature in this test and kind of follow-up stuff from where you've come so far.
Mm-hmm. Yeah, so we're also looking at the genetics. So not just at this one receptor, we're looking at the whole entire genome, because one, that will tell us, okay, what is the status of this one receptor? But also two, is we can look at population genetics of Varroa. I think based on some other research that I've been seeing, is that they can differentiate quite rapidly, because they tend not to have a lot of migration into, there's not a lot of heterogeneity in populations. So that means that they're isolated and they can rapidly evolve. So that's what we're doing with these 500 genomes. We just got some more money to do some others as well. So hopefully we'll be able to find out, are all Varroa in the United States the same, or are they all pockets of difference based on where beekeepers are and how they move around. So I think that's gonna be really important to find out, can we manage Varroa as a single unit across the country? So we're also looking at new formulations of either amitraz or other products. Actually, I'm actually going to go out the door here and do another test on this one new product that we got our hands on. So we're always looking for constantly evaluating new products. I think it's really important also too for beekeepers to be aware of what works and what doesn't. So check with other beekeepers in your area, check to see what the scientific literature there is to support these new products, because I just want to make sure that everybody is buying something that actually works and is effective against Varroa, but also safe on the bees. So that's another critically important factor here as well. So yeah, new product development, but then also too, we're trying to come up with crazy weird things. So we're trying to feed these bees algae that express double-stranded RNAs to kill Varroa. We got some good preliminary evidence so that, yeah, we can use that system for delivering something to kill Varroa. So there's so much more work to do. It'll never end.
Yeah.
I'm gonna be doing Varroa research for the next 20 years. So
well,
I'm not complaining, because it's a great field to be in.
I feel like we're in good hands. So thank you for the work that you do. And I think you also did some work a few years ago, and maybe the paper's coming out in the future, about the tremendous impact that Varroa has on the health of our bees.
Yeah, so there was a number of papers that we came out. So last year we published this one where we looked at a migratory beekeeping operation and used two different stocks of bees. We used the Varroa sensitive hygienic stock that we have here at the lab called Pauline, and we compared that to an unselected commercial stock. And what we found out was that Pauline that was a result of this intense, about 20-year breeding program. We found out that when it's used in commercial beekeeping operations, they tend to have similar populations of adult honeybees. They produce similar amounts of honey under good conditions. That's the caveat of under good conditions. Bad honey years, that difference gets pretty big. And that's something that we know. But they also have significantly higher survival rates, lower virus loads, and mostly due to low Varroa populations. So in the field, side by side, we could see that just controlling for Varroa, your colonies survive at a much higher rate. That's a major part that we've been finding out. That was something that we just published we had one paper that came out last year, and then we have another one that we're going to resubmit using multiple years of data and other data points as well.
Good.
I
Look
also
forward to
submitted
that.
another paper where we were in Iowa, and we did a 2-year study looking at the effects of pesticides and Varroa on bees. So we collected pesticides from pollen and measured the pesticide exposure. So to each individual pesticide, but then also to these composite measures, along with Varroa. And so the first year we didn't treat for Varroa, and guess what happened? Varroa populations went really high. We lost 75% of our colonies. Second year we came back, we treated for Varroa. We only had I can't remember the numbers off my head, but something like 15% colony losses. That's pretty good. The really interesting thing about that, though, is yeah, we could find out, okay, Varroa is a big important factor. What about pesticides? Well, it turns out in that second year where we had 15% losses, we had significantly higher pesticide exposure than the year before. So pesticides are definitely a part of this. Don't get me wrong, but I think that the effect of Varroa is so much bigger that let's focus on Varroa first, and then we can think about pesticides, because it has they can have an effect, but I think that without controlling Varroa, it kind of skews the data.
Yeah, I agree. That's kind of my message to the beekeepers that I work with is like, yep, pesticides can be an issue, but let's worry about the things that are in our control. Let's handle mites. Let's make sure we manage queen events. Let's handle nutrition, make sure they don't starve to death. Let's don't feed them too much. And that really takes care of a lot of problems. Although there can be pesticide issues now and then, but it certainly doesn't account for most of the bad stuff that I see as an apiary inspector. So
yeah,
handle the stuff that we can handle.
Exactly, because we've known pesticides are bad for bees for 70 years now. And I'll tell you what, they're never going to stop applying pesticides,
Right.
whether it's on farms or whether it's for mosquito control. Pesticides are here. We have to accept that. That's the future. We can ban a few here and there, but it's like whack-a-mole. You ban neonics, something else comes in and replaces it.
Yeah.
They're always going to be around. So let's focus on things that we can. So things like Varroa is definitely a good starting point. But it's also too, we have to really pay attention to which pesticides we're studying, because again, there's a lot of focus on neonicotinoids, and yes, they are incredibly toxic to bees. Yes, they can result in high bee kills, especially early in the season when the disease are going around. There's no question about that. However, I think we have to start looking at some fungicides though, because we have some really interesting data that shows that these fungicides that are completely non-toxic to bees, whether it's in the field or in the lab, they can withstand very high concentrations of it. It's not going to have those toxicity effects, but it can affect their physiology in other ways. So, for example, the more fungicides they're exposed to, the lower their vitellogenin levels are. Vitellogenin is this, it does everything. So,
Right. So,
when I say vitellogenin, just think it does everything. And
right.
what we find out is that when they're exposed to more fungicides, their vitellogenin level decreases. And it may have an effect on their ability to control Varroa. We just did an experiment here at the lab where we fed them a fungicide, and they look fine throughout the season. But then when Varroa populations go up, the ones that are fed this fungicide, Varroa goes higher.
Yeah. So the other thing I'll say to beekeepers, it's complicated, right? So, well, if you put fungicide on an adult bee, it doesn't really affect the adult bee, but there are other impacts, and there are so many moving parts, and it really is complicated. I wish it were easier, but it is difficult.
Yeah, there's a lot of heterogeneity in the landscape when it comes to pesticide exposure, because we found it in this one apiary over here. It doesn't mean this apiary over here has the same exposure. It's so patchy because it depends on mostly what crops are growing around it. And so, yeah, it's hard to make a broad general statement about pesticides, how it's going to affect honeybees across the country in every situation. It is situational. So, for example, in almonds, when they spray those, they do spray a lot in almonds. Yeah, we can definitely see acute mortality incidents in almonds when they are sprayed. There's no doubt about that. However, when it gets like the low level sort of things, it's much more difficult to find those effects, mostly because the effects are subtle. So you need huge populations of colonies. So yeah, I would say you need hundreds of colonies to actually assess those impacts because they are very subtle.
Yeah, cool. Thank you so much, Frank. I really enjoyed talking to you this morning. And again, I really do appreciate all the good work that you and others at USDA and other researchers at our land-grant universities do for our honeybees. And as always, if I can help, please holler at me. I really do enjoy working with you and appreciate you.
Yeah, again, I always know that I can't, this project is mostly a collaboration between us. So yeah, let's keep it going. And then also so really important too, that beekeepers have to know that this is a two-way street too, that
Yeah.
you have to work with us and we have to work with you. So I love working with beekeepers. Most of the big field projects we do are in collaboration with commercial beekeepers. So those opportunities for research are really important for making sure what we're doing actually means something to the beekeepers.
Great. All right. Well, have a good day, Frank. Keep up the good work, and we'll talk to you soon.
All right, good to talk to you, Lewis.
All right. See ya.
Anytime.
All right. Bye.
Bye.